Okay, in this video we're going to take what we learned about this diagram. We're going to just sum it up in some nice clean bullet points to help those of you who learn better off of the short list and the less talk. We have four phases essentially. We have depolarization, we have plateau or maintained depolarization, we have repolarization, and we have a refractory period. Depolarization starts with that cardiac cell having a stable resting membrane potential at negative 90 millivolts. We're going to see excitation spread between those fibers through gap junctions in the intercalated discs. Depolarization occurs because we have some fast sodium channels that open up and we have sodium flooding into that cell, and that sodium flood is what is the cause for our rapid depolarization. Those channels will close spontaneously within milliseconds, and at that point we're going to move into our plateau or our maintained depolarization. It lasts about 250 milliseconds. Plateau in a regular neuron is going to be about 1 millisecond, but plateau here in the cardiac cell is going to be about 250 milliseconds. We're going to see slow calcium channels open. That's going to allow calcium to flood into the cytosol. We're going to see potassium channels close. Calcium is going to bind to the troponin. It's going to allow for that actin-myosin cross-bridge formation that you learned about in sliding filament, and we're going to see tension develop within those fibers. Repolarization is our third step. At this point, calcium channels are closing, potassium channels are opening. We're going to move from that depolarized state up around the 20 millivolt mark back down towards the negative 90 millivolt. That's going to happen because no more calcium is flooding in and we have potassium leaving the cytosol of the cell. Our fourth and final stage to talk about here is the refractory period. The key that we talk about here in contractile fibers with refractory period is that refractory period is very, very long. It's much longer than the contractile period, and because of that length, the heart can refill, the chambers can refill, and the side benefit of that is that we do not see cardiac tetany. Alright, so let's step into ATP. All of this work is going to need a ton of ATP going on. So where are those cardiac cells going to get their ATP? And they're going to get that ATP from aerobic cellular respiration. If you don't remember aerobic cellular respiration, it was covered extensively in Biology 181. You may or may not have had some of it in 201, just depends upon who your instructor was, but there certainly is a decent section in your textbook on aerobic respiration, cellular respiration for ATP production. Remember that cardiac muscle itself is able to produce a very small amount of ATP from creatine phosphate. Creatine kinase, CK, or creatine phosphokinase, CPK, in the blood is an indicator of muscle damage. So when we look at blood chemistries and we see an elevated CK, or depending upon the lab, a CPK, just depending upon what they're testing for, we may have an indication for cardiac muscle injury. That could be caused by a myocardial infarction. But remember that CK and CPK can happen with any muscle damage. So you could have easily have stubbed your toe, slammed your thumb in the door, be a klutz like me, and fall down, over a crack in the floor that is otherwise known as a seam between tiles to other people. So you can't rest a diagnosis of myocardial infarction on a CK or CPK result. We're going to stop this video. It's a relatively short one, and then we're going to pick up with a discussion on the electrocardiogram.